Hello everyone, guys. So this is the first lecture that I'll be recording here. So this is the lecture where we were gonna discuss the steps through which the genetic information, which is stored in the DNA of living organisms, it's converted into protein through the two processes named transcription and translation. Now, I'm gonna divide this lecture, first of all, into two parts. In the first part, I will discuss only transcription, and in the second part, I'll discuss translation. So it's gonna be a little bit easier for you to divide the learning. I'm also gonna be skipping the first few slides here, which dealt with the first experiments, which indeed has shown the relationship between the proteins found in living organisms and the presence of specific genes coding for these proteins in the DNA of the same organism. I will not ask you about these experiments in the midterm test. So what we're going to be focusing here ultimately is the second step of what is called the central dogma, which is that basically that dogma, that knowledge that is shared by scientists, which deals with basically the passing of the genetic information from one organism to the other, and that's what we discussed by talking about DNA replication. But most importantly, it deals with how the information stored in the DNA of living organism is translated into protein through the same two processes in all living organisms. The first one being called transcription, which is where the information in the genes is copied into a temporary copy of the same gene, referred to as messenger RNA or mRNA, and this is done through the process called transcription. And the second step is where the mRNA that's being produced through transcription is then translated into protein. So the temporary information stored here, it's used at the ribosomes through the process called translation to produce a specific sequence of amino acids joined together, therefore a peptide. So let's start with transcription. So these are the two steps that we were talking about here, right? Mentioned before. So when we talk about transcription, again, we're talking about that process in which the information stored in very specific region of the chromosomes of the DNA called genes, it's copied into a temporary RNA copy referred to as messenger RNA. This is done by copying only one of the two strands. So DNA comes as a double helix with two antiparallel strands. So only one of them gets a copy. And this is done by an enzyme which is similar to the enzyme that we mentioned, or those enzymes that we mentioned when we were talking about DNA replications, which are DNA polymerases. So enzymes in this case are referred to as RNA polymerases because they make RNA, which do attach together nucleotides, in this case ribonucleotides, by attaching them by forming phosphodiester bonds, and by attaching the nucleotides to the 3' of the previous nucleotide. For this reason, they get the RNA in this case too to grow in the what is called the 5' to 3' directions. In this case, in this process, since we are dealing with the RNA, so obviously when it comes to the nitrogenous base, we don't have thymine present there, but we have uracils. And when it comes to transcription, conveniently, transcription can be described as 3 different steps, which in reality, they happen in continuity with each other. So the first step is referred to as initiation, the second one as elongation, and the third one is referred to as termination. And these steps somehow, if you want, mirror the organization of the genes. So if you look at any gene, so any of these coding sequence, it would have, first of all, a region referred to as the promoter. As the name says, this is the region that promotes the transcription of the gene. So that's the region that basically turns on or off the expression of the mRNA, the formation of the mRNA. And then right after this promoter, in continuity, there is the transcription unit, which consists of 2 different parts. You have what it is, the sequence that basically contains the coding regions, plus minus other sequences called introns. And this is the sequence which then later on will be translated into proteins. And then there is a final sequence here, which is right after the coding region, which is referred to as the termination sequence. So these two sequences are referred together as the transcription unit here. So the three steps that I mentioned before, initiation, elongation, and termination, refer to where they basically take place here. Initiation takes place in the promoter. Elongation takes place with the copying of the transcription unit until RNA polymerase reaches the termination sequence here, where termination, the last step, takes place. Okay. So now, moving on here. So first phase, initiation. So this takes place at the promoter. Promoters are sequences of DNA which vary in length. So every gene would have one, and they go from 200-300 base pairs all the way to 2,000, 2,500, 3,000 sometimes base pairs as well. Their function, as we said before, is to turn on and off the transcription of a gene. Why are they necessary? They're necessary because if you think in your genome, if you take your own DNA, for example, you will see that only 1 to 2% of your DNA actually corresponds to coding genes, so genes that give origin to proteins. The rest has different functions, which we will not talk about here. So now, obviously, for transcription, now you don't want to transcribe the whole DNA if only 1 to 2% has to be translated ultimately into proteins. You want to focus only on the necessary sequences, therefore the genes. So you need to have regions where you signal the enzyme RNA polymerase to go to and to start transcribing. So it does not transcribe the rest. The same point is for the termination sequences. They tell also RNA polymerase where to stop. So RNA polymerase polymerizes nucleotides only where it's necessary and does not waste energy ultimately to copy regions which are unnecessary. So when it comes to the promoter here, so these are sequences of different length, and basically what they have in terms of function is to recruit the enzyme which then later on will copy the DNA into messenger RNA, which is RNA polymerase 2. This recruitment is done through other proteins called transcription factors. So transcription factors, so this is where the promoter will be here. So transcription factors are other proteins that do basically bind to the DNA here in very specific ways. Okay, so there are different transcription factors depending on the gene you're talking about. So if a gene has to be expressed only in your muscles but not in your neurons, for example, you would have certain transcription factor binding here. And the reason that they know where to bind is because they bind to very specific sequence of DNA. So a gene that has to be expressed in your muscles would have only these sequences which attract transcription factors that do activate the expression only in muscles. If that has to be expressed another gene in neurons, it would have other sequences bound by different transcription factors, which then turn it on in a different tissue. So in this case, neurons, different cells, in this case, neurons. Okay, so the promoter, all it does is basically you have transcription factors binding specifically to the promoter and therefore recruiting the polymerase, RNA polymerase here, right at the beginning of the transcription unit. By doing that, basically it's setting this gene to be ready to be copied. But the last step in all of this is the binding of RNA polymerase will lead to the partial opening of a sequence right before the transcription unit here. This opening looks like a bubble and therefore is referred to as the transcription bubble. It's a very small region, about 12 to 14 base pairs. Its opening is facilitated by a sequence here, oftentimes, which is referred to as Tata box. So it's a sequence that is very rich in thymine and adenine. So sequence where you have that the two strands are joined together by fewer hydrogen bonds compared to sequences that are rich in G and C's, guanine and cytosines. So it makes it easier to open it up. So summarizing here, in the initiation step, what you have, which takes place at the promoter, all you have is that basically that the entire machinery that turns on transcription is put together at the promoter. So this is done through transcription factors that bind to specific sequence and they recruit the RNA polymers, and this leads to the opening of a transcription bubble here, which then will give access to RNA polymerase to the specific sequence of the one of the two strands that is supposed to copy. Okay, obviously the template, the RNA polymerase is the one that's going to be copied here, will be the one that goes from 3' to 5', because RNA polymerase, as we say before, can polymerize only in the direction of 5' to 3'. So once this is done, initiation is done and elongation takes place. Okay, elongation starts at the very first nucleotide. It's called the transcription start site, so it will be named as +1 transcription start site. That's where the first nucleotide is put complementary to the template strand. As you can see, the template strand here is from 3' to 5', and depolymerization done by RNA polymerase is done from 5' to 3' by joining different nucleotide here. So this is already part of the elongation process that starts here. So now when elongation is taking place, so RNA polymerase is adding all these ribonucleotides together complementary to the template strands, exactly like in DNA replication. The only difference is that RNA polymerase does not need a primer, unlike DNA polymerase 3. You remember DNA polymerase 3 needed a primer to start copying. In this case, RNA polymerase 2, which is an RNA polymerase like primase, does not need it. So it starts at transcription start sites and basically keeps going through the template sequence, copying it into messenger RNA. When it's doing that, basically you would have that while this is copying the template sequence, the sequence that's already been copied will start closing it up, whereas the sequence that is ahead of where the DNA polymerase is going to will keep opening up. So by doing that, basically what it looks like is that the transcription bubble, so that region that it's open, this region here, the 12-14 base pair region here, looks like if it's moving through the sequence, the sequence that has to be copied in the gene. And so this process, very simply, elongation, is nothing else than the process of polymerization of RNA polymerase 2, which forms basically a copy of the template sequence in the gene. And this goes on until RNA polymerase reaches the termination sequence, which is where the last step, termination, takes place. So this is done in different ways in different genes. A very common way, and that's the only one that I'll describe here, is done by having basically this sequence present at the end of a gene. So many genes do have a sequence similar to this one right here. If you look at this sequence, so this will be the DNA sequence, okay, present there, which then you would have that this template sequence, that 3' to 5', would then be copied by RNA polymerase into RNA, and this is the RNA sequence that would come out from this. If you look carefully here, so what you can see here is that when RNA polymerase is copying this sequence, it's basically producing two sequences, here in yellow, this one right here, and here, which are basically anti-parallel with each other. Not only that, but past that, is also going to add a series of uracil. Uracil will be very weakly bound to the corresponding template, the nucleotides in the DNA sequence that's being copied at that time. So what it means this, and how does this cause termination, is that imagine the RNA polymerase moving in this direction. So you start copying this sequence here, this sequence here, this sequence here. Now, as soon as this sequence is copied, these two sequences right here, which are anti-parallel to each other, they will form a partial double helix here. So what they will form with a loop in here, this is referred to as a hairpin. They will form a hairpin, which then will actually pull away the remaining sequence that is copied here too, which is very rich in uracil and therefore weakly bound to the DNA here. So this pulling away caused by the formation of this hairpin caused basically the separation of the RNA and the RNA polymerase from the DNA template. This is done together also with some proteins that are actually helping this process. But overall, it's the pulling away that caused the separation of the growing messenger RNA and the RNA polymerase from the DNA, and therefore the termination of the transcription process. So this is how basically a gene is copied, first through initiation at the promoter, then there is elongation, and then finally terminations. So at this point, the RNA has been formed, which is identical or complementary, if you want, to the template DNA present in the gene. Now, it's important to understand that that is not the final product of the, that's not the final messenger RNA, which then is going to be copied into a protein. And we'll talk about that in a second. First of all, though, we have to discuss one thing. So now, this process we just described takes place for protein coding genes, but also can take place for non-protein coding genes, which give origin to RNA like ribosomal RNA or transfer RNAs, which are necessary for a cell to survive. Now, there's a big difference between prokaryotes and eukaryotes in this sense. In prokaryotes, basically, whether a gene is coding for a protein or is not coding for a protein, they work in a very similar way, and in fact, they are transcribed by the same RNA polymerase. In eukaryotes, instead, they have different promoters, which are recognized by different transcription factors, which then they will recruit different RNA polymers. So in eukaryotes, RNA polymerase 2 is the one that would copy the protein coding genes. Whereas for tRNA genes, and one of the 4 ribosomal RNA, there are 4 of them, so it will be RNA polymerase 3 that transcribes them. Whereas for the remaining ribosomal RNAs is RNA polymerase 2, polymerase 1, that would do that. All right. And again, these are recruited because there are different promoter sequences present in these different type of genes. So, as we say before, now RNA polymerase, we're focusing here only on protein coding genes, has copied the template DNA into an RNA. Well, that RNA is not the final product, which then will go into the ribosome. Before it gets to the ribosome, it actually has to be matured through 3 steps, which all together are referred to as post-transcriptional modifications. So there are 3 different steps. But before we talk about that, we have to understand how it will look, the immature RNA that's produced by transcription. So this will be ultimately, so the promoter will not be part of it, but this will be the RNA that would have been, or the DNA that would have been copied into RNA. So these RNA would have, first of all, a region in the front which does not contain the information to produce the protein. The same thing would have a region on the back referred to as the 3' UTR or untranslated region. So this was the 5' untranslated region, which similar to the 5' untranslated region, the 3' untranslated region gives origin to, does not give origin, to any protein. The protein coding sequence is contained between these 2 different regions, but is not copied directly all into a direct singular piece. It's contained by sequences called exons, but in between the exon, you still have remaining sequences which do not code for the amino acids. And therefore, for these mRNA to be ready to be copied into protein, these sequences would have to be removed. Okay. Finally, there's also what is called a polyadenylation signal here, which is involved later on for adding stability to the RNA. So the RNA that's formed, first RNA, the immature RNA, has to go through 3 different steps of modifications post-transcriptional. So what are these 3 steps? There is the first one is referred to as the addition of a 5' cap or 5' capping altogether. There is the removal of those intron sequence here, non-coding sequence that referred to as intron splicing. And the third step is referred to as addition of a 3' polyadenylation or 3' capping by polyadenylation. Okay. So let's look at the 3 steps. 5' capping. So this is an important step, which is done for 2 main reasons. First of all is to protect the 5' of the immature RNA at this point from degradation. RNA is very unstable. It would easily be recognized by some nuclease here and degraded very quickly if it was not capped. So, and this cap is basically done by adding a guanine containing nucleotide that basically is reversed and attached to the 5' here. Similarly, on the 3', you have that the degradation in this case is prevented by adding not a reverse nucleotide, but a long stretch of adenines, basically. So this stretch can go from 50 to 250 adenine-containing nucleotides or ribonucleotides, which will be attached here on the 3' where there is what is called the polyadenylation sequence. And so in this will look, that's how it looks, the polyadenine tail. So we have already two modifications here, 5' capping for protection, and also I forgot to tell you, this is also important to signal this RNA to get to the ribosome as well, to be carried to the ribosome. And this polyadenylation, poly-A tail, which is important for preventing degradation from the 3' end. Now, the last step in the post-transcriptional modification is the removal of introns by splicing. So now, this splicing can be done in different ways. So it can be done through the help of different proteins. So proteins and RNA complex, which are referred to as short, small nuclear RNA, and there are complex with proteins there as well. And some introns actually they do splice themselves on their own without the help really of proteins and short small nuclear RNA complexes there. So some of them they do splice on their own. I don't know if you remember, but when we were talking about the origin of life on Earth, we were talking about the fact that people believe that originally the enzymatic activity was done by RNA molecules and not proteins. One of the supporting evidence is exactly done by the way some introns are spliced on their own, showing that the RNA molecule, the immature RNA molecule, can splice itself and therefore can have enzymatic activity on itself. So this process, which you don't need to remember in detail, is done either by a complex of protein and short small nuclear RNAs, or is done by the intron itself, depending on the immature RNA we're talking about. In the end, no matter how it's done, basically what this done, it leads to the removal of the intron here, which will form a circular molecule, which is referred to as the lariat, and the joining of the protein coding sequences, which are referred to as the exons. So this exon 1 here, after the removal of this intron, will be joined directly to the exon 2, which again includes the information for the protein. If there is another intron here, will be removed as well. Now, this is actually a very interesting process. The reason is, first of all, it's done only in eukaryotes, not in prokaryotes. Prokaryotes do not have introns. Eukaryotes do have it. Second thing is that this process of removal of introns, so you have to imagine some genes do have multiple introns in it. So multiple exons and multiple introns in between. But the way in which these introns are removed, and so the way in which these exons are attached together, can be different from one tissue to another one. So it can be different, for example, in skeletal muscles, so in your muscles, and in your neurons, in your brain. So the combination of exons that you can have can be very different. And so for these reasons, what you have is what is called alternative splicing. So one example here is, for example, alpha-tropomyosin here. So this is spliced in different ways depending on the type of muscle that you're talking about. So there are three main types of muscles that you have. You have skeletal muscles. These are the muscles that basically move or attach to your skeleton and are involved in motion, mostly voluntary muscles. Then you have your cardiac muscle, which is the one found in your heart, which pumps blood around. And then you have smooth muscle, which is an involuntary type of muscle. By involuntary, I mean you cannot really control it. And it's found, for example, around your guts, around your blood vessels, and controls vasodilation, vasoconstriction, and so on. And so alpha-tropomyosin is expressed in these type of muscles, but it's expressed differently. If you compare, for example, the way it's expressed in smooth muscle compared to skeletal muscle, you will see that the exons are not all the same. So this picture gives you a better idea. So this is the smooth muscle here. This is your skeletal muscle. And so this is your immature RNA, so the one that will contain the exons, labeled with a number, and the introns here, which are in green. So these introns have to be removed. But what you can see here, for example, in the form that you get after splicing in the smooth muscle, you have that the sequence is exon 1, exon 2, exon 4. There is no exon 3. The same thing for exon 10 and 11 are not present in there. If you now look in the skeletal muscle, now you have that instead you don't have exon 2, but you have exon 1 and 3. Not only that, but you don't have exon 12 as well. So as a result, now if you think the same identical gene, so this is the same gene that works both in smooth muscle and skeletal muscle, what has led to is the production of two very different proteins, one found only in the smooth muscle and the other one found only in the skeletal muscles. And that's the reason why, if you look at how many proteins, for example, you produce in your own body, we're talking about 100,000 proteins. But if you look at the number of genes that you have, it's about 20 to 30,000. So, and that's possible exactly because some genes are giving origin to immature RNA, which are spliced alternatively in one tissue versus the other one, therefore producing more than one single protein. So, as a result, summarizing all of what we discussed here, through the process of transcription, RNA polymerase has produced an RNA sequence, which is first the immature RNA, which is a copy of one of the two DNA sequences present in the transcription unit of a gene. And then, now this RNA goes through 3 steps, through post-transcriptional steps. So there is a 5' copy, a 3' polyadenylation, and then the removal of introns, which can happen differently in different tissue through alternative splicing as well. And then this has led to the formation of a mature RNA, messenger RNA, which now is ready to be copied in the ribosome into the protein. And I will stop here for this lecture. I will record translation in a separate lecture later on. Okay.